Good morning, everyone. My name is Nashwa Al-Gindi, and my presentation topic today is about quality consideration for first generic Tiotropium Bromide capsule-based dry powder inhalers. This presentation will deliver several learning objectives. We will explore the general constraints in dry powder inhalation, formulation, and manufacturing. We'll discuss potential development challenges for capsule-based dry powder inhalers to improve the quality and the performance of these products. Then I will share valuable recommendations for highlighted product characterization studies, and we'll end it with focusing on general quality considerations for primary drug product stability batches. First, I would like to give you a brief introductory about DPIs. There is a lot of many classification for DPIs. However, here I will focus on one classification based on the device constituent part, the dose container. DPIs can be meter dose inhaler, which a powder reservoir contain multiple units in one container, or pre-meter dose inhalers, which are plasters, discus, cartridge that contain multi-unit dose inhalers, or capsule, which is single unit dose inhaler, and this is the focus for our talk. As a fact, generic DPI can be more complex to review compared to meter dose inhalers. Also, plasters and capsule DPIs-based DPIs are the main type submitted to FDA since 2015. So the reference listed product, the RLD for the first generic capsule-based inhaler is Priva inhaler, NDA 021395 by Boehringer Pharmaceuticals that has been approved in 2004. The first generic Spriva inhaler is Loop inhaler by Lupin and 211287 that has been approved in 2023. Each capsule of this drug product contains a dry powder consisting of 18 microgram of the API, tiotropium, blended with the carrier lactose monohydrate. There is the product-specific guidance, draft product-specific guidance with this drug product has been published in 2023 and includes many recommendations for BE studies. One of these recommendations is that the test and the reference product recommended to be Q1, Q2 the same. Also, the test and the reference product, the device should be similar in the mechanism, the breath-actuated pre-meter single unit dose capsule-based format. Also, the number of the doses, the size and the shape and the resistance, the external operating principles and the critical design attributes. There are in vivo PK and comparative clinical endpoints recommendation in this PSG. However, I will focus my talk here about detailing about the in vitro BE studies. For the in vitro BE studies should be conducted on 3 exhibit batches, 10 units per batch for the test and reference product. And just here the table summarizes the 2 in vitro studies for in the PSG for this drug product, the single actuation content with the SEC and the aerodynamic particle size distribution APSD. And here in the second column, the design for each study and the summary of the BE analysis. Here they are very related to the quality tests, the SEC, similar to the dose content, delivered dose uniformity and APSD from quality perspective. Then I will jump here into the composition of the capsule-based DPIs. So usually the composition table for sure is usually submitted in the module 3 in P1, which contain the ingredient, quality standard, function, quality, quantity, milligram per unit, and the concentration % weight per weight. Firstly, the API here, the quantity should be per the RLD labeling, and this is the DPI strength, which is the amount of the meter, the amount of the drug from the capsule, not the target delivered dose, which is the mass of the drug delivered from the mouthpiece. Then the carrier, and it should be here based on the PSG for this specific drug, it should be Q1, Q2 the same, and this is the most preferred for most complex product. Then the capsule size and composition, and the capsule shell material may be in some cases different between the RLD and the test, and it may be acceptable or may be adequate in some case. Here in our situation, the listed product is gelatin material, however, the first approved is HPMC material. They are different in moisture content. However, it could be acceptable to use different material provided adequate justification with supporting data. For example, study to demonstrate that the fragments generated by piercing and actuation of the rupturing of the capsule are not inhaled by the patient. Also, also the ink composition should be listed in the composition table. Then I will start with one of the first topic in my presentation about the learning objectives, which is the general limitations for DPIs in general. There is a lot of constraints. Firstly, when we look at the formulation design, as we said before, Q1 the same. Q2, this is preferred. For this specific drug product, yes, it's in the PSG highly recommended. However, for some applicants, they may come up with different Q2. In this case, rationale and adequate justification should be provided, as well as pharmaceutical development data for in vitro testing or studies for a combination of the between different ratios of the drug to excipient, which including combination above and below these ratios for the RLD and test. Justification is really important here, and data. Also, average. Generally, it discourages per ICHQ8 guidance. However, if it's used, justification should be also provided. For example, drug loss during proposed manufacturing process. Another thing is the manufacturability, and for sure this is powder. Powder characteristics are very challenging. So many powder should be tolerated, should have a tolerated particle engineering. The shape and the surface characteristics of the powder can affect the aerosol performance and the airflow, and as well as the drug release. Another constraint for the DPI, specifically for the capsule base, is the device. And as I said, per the PSG, the device design is very important to be similar with the RLD design. Also equivalent design, also the operating principles and the resistance. This is very critical attribute for the device. Now, my first challenging question, to keep you in the loop, which of the following examples can justify the proposed average in DPI? A, drug loss during proposed manufacturing process, drug loss due to degradation of the drug substance, drug loss due to deposition on the device constituent part through unit life. The answer is A. Now I'll go deeper into the challenging in developing generic DPIs. To develop a generic DPI with a consistent DPI performance, which represented by the delivered dose uniformity, for example, and APSD. A lot of unique challenges are related to the drug product formulation, API and excipient, manufacturing process, device design characteristic, and ended with the user interface considerations, which is the patient handling. Now, a couple of examples about the formulation development challenges. Firstly, the risk may come from recrystallization of the micronized material, which could lead to particle growth, uncontrolled particle growth during shelf life of the product, thereby affecting the product quality attributes, specifically APSD and DDU. And APSD and DDU are considered the key performance characteristics and measures in assessing BE studies. And accordingly, it also in turn may affect the in vivo performance of the drug product. Also, missing proper control on critical material attributes of the drug constituent parts of this combination product, like the API or the carriers, without proper justification. One of the big challenge here is the amorphous content of micronized particles. Okay, mitigation. What is the control strategy to mitigate this risk? Include equilibration period or condition to allow the recrystallization to occur prior to any additional manufacturing unit operations. Also, consistency of the manufacturing process. Also establish particle size distribution specification based on the actual data from the micronized lots used in the manufacturing of the drug product exhibit batch or primary stability batches, and ended with continuous in-use control, which is the routine control of the critical material attributes. Another risk here, based on the diagram I can show you here, this is, sorry. So based on this diagram, you can see the powders, the DPIs consisting of API particles, very small, usually micronized within 5 micron, attached to the surface of the carrier to form agglomerates. This agglomerate fluidized to form a separate agglomerates once it's subjected to the airflow with the passive DPI, with the inhalation by the patient. It starts to be de-agglomerated to disperse the API particles out of the surface of the carrier to be inhaled into the site of action. So the carrier here is very critical component. So the carrier grade may lead to particle size growth over time during storage. Also, the carrier morphology, shape, and surface roughness may affect the adhesion force between the API particles and the carrier particles and affect the aerosol performance. Mitigation control strategy here is to adjust the ratio of the fine to coarse grade particles if they are there, because if the fine particles can be attached to the coarse particles and may affect the attachment or the adhesion of the API particles. Also continuous in-house control for particle size and morphology. I will jump here into manufacturing challenges. After formulation, we go into the another challenge with the manufacturing, and I will focus in 2 challenges. Firstly, the blend uniformity. Blend uniformity, because this drug product, the challenge here is the excipient over 90% of the whole formulation. So there is a low drug load. Also the flowability due to excipient lot-to-lot variability. How to mitigate these risks with the control strategy? Firstly, how to select the proper blender type, the equipment used. Control the critical process parameters like the blending time, speed, blender fill level. Also the excipient critical material attribute. Control the environmental conditions, conditioning, hold, equilibration period after blending. In addition to the final blend characterization, which includes the uniformity, bulk density, and particle size. Powder fill, another challenging during manufacturing. Fill weight variation and segregation of the API and the excipient lead to failure in the APSD, DDU, and content uniformity. Also, the moisture content, very critical for affecting the APSD and DDU. Mitigation control risk here. It's important to identify the critical process parameters and characterization of the fill weight, content uniformity, moisture, APSD, DDU, and this is from the filled units through stratified sampling during development. Highly recommended. My last challenge here about the container cluster, which is the big deal for combination products. In this specific product, capsule-based product, it has the device, it has the capsule that containing the powder, and the plasters, which is the primary packaging. Here I will give you some recommendations important for this device combination. Firstly, for the capsule dimension is critical for the adequate insertion of the capsule into the inhalation device, piercing, and drug delivery. So it's recommended to include the capsule dimension in the drug product specification, which is the lock length. What about the blister foil and the overwrap pouch of the device? They provide the protection from moisture permeation to the blend formulation at in use and shelf life. So it's recommended that the functional layers of the foil should be controlled for the thickness of the individual functional layer, that to assure the consistency, the consistent quality and performance of the primary and secondary packaging. Also for the device, the inhaler device specification should include the critical functional parameters, and I listed here a couple of these functional parameters to be included in the specification. Also, another recommendations. For the needle diameter, this device has a needle and a mesh, which used for inhalation of the dispersing of the powder out of capsules. We recommend the applicant to investigate the potential effect of the needle diameter and the mesh material on the product performance and powder residue in the capsule after testing APSD and DDU. For the button push in force, it's recommended to be determined over the lifetime of the inhaler containing the capsule in the chamber for consistency. And in some cases, some applicants like to also do the same testing without capsule to see the differences. The cap opening force, which is also critical, especially for patient handling. It's important to demonstrate if this force changes over the use of the unit, of the use of the device, and if there is if so, these changes, whatever it's reducing change or reduced force, is sufficient to keep the cap closed and open by the patient easily. My second challenging question: which of the followings are considered formulation development challenge in DPI? Fill weight, B, excipient rate, C, device resistance, D, amorphous content of sprayed APIs. The answer is B and D. Now, my third topic here is about the drug product characterization studies. Per the revised draft FDA guidance on MDIs and DPIs that has been published in 2018, there are a couple of characterization studies should be conducted on the DPIs as a one-time study to demonstrate the robustness and the performance of the product and to support the labeling instruction. Here in the right hand side, you can see I listed the characterization study that can be conducted, should be conducted for capsule-based DPIs. In general, DPI products units used for the characterization studies should be stored under conditions consistent with the storage conditions identified in the label. Also, to support the proposed shelf life at the intended storage condition, it's recommended that some characterization studies should be performed at the beginning and near to the end of the shelf life of the product's units, including in use period, cleaning instructions, and robustness. Now I have some recommendation for a couple of characterization studies. Firstly, for the robustness, this is based on this specific product capsule-based DPIs. For robustness, it's recommended to assess the physical robustness of the capsules following the dropping and or vibration of the blister card for any damages. In the cleaning instruction study, it's also recommended to determine the drug residual at the end of the experiment for each unit, in addition to visual inspection for any capsule fragments and particle residuals. Fragment study. This is really critical for capsule-based DPIs, especially if the applicant is using material different from that of the RLD. So assess the puncturing and fragmentation behavior of the proposed capsule in comparison to the capsule of the RLD. Here is in this case, it's HPMC versus gelatin. Even if the applicant using gelatin, it's recommended to still perform this study to confirm there is no any fragments that can be inhaled by the patient. My last recommendation for effect of use study, where we are recommending the applicant to inspect the partially used product units after coming from clinical studies for any external damages. In addition, to evaluate the functionality and piercing mechanism of the clinical return inhalers for any mechanical defects or failures. Like, for example, the lack of rattling of the capsule, how to pierce the capsule, failure of pressing the button. This is really critical functional defects. My last challenging question here: which of the following drug product characterization studies should be conducted on the DPI product units at the end of near the end of shelf life? A, cleaning instruction, B, temperature cycling, C, robustness, D, in-use period. The answer, A, C, and D. I would like to end my talk with a general quality considerations for the primary DPI stability batches. Firstly, for the drug product constituent parts, the API, excipients, and device. It's highly recommended to use 3 different lots to manufacture the 3 primary stability batches. Drug substance discrete lots, the device constituent part components, as well as the critical excipients. And here, this is the carrier. For the batch size, given the complexity and the potential risk involved in scaling up the manufacturing process for DPI products, it's highly recommended to submit stability data from 3 exhibit batches at the proposed production scale. However, if this information is not available at the time of submission, it may be acceptable to provide at minimum 1 batch at the proposed scale and commercial scale and to 1/3 of the commercial scale. However, if this is the case, it's highly recommended for the applicant to provide in-process and batch analysis data from several batches that's manufactured using the commercial proposed manufacturing process, like engineering, like developmental batches, performance qualification batches, to confirm the robustness of the proposed manufacturing process. At last, the product characterization studies should be conducted on the to-be-market configuration and the version of DPI products, also 3 batches with reasonable sample unit to support the reliability, reproducibility of the product quality and performance. And here are the resources that can help with the DPI products: the FDA guidance, USP Chapter 5, and USP Chapter 601. At the end, my closing remarks here, again, DPI products can be more complex to review than MDIs and other complex products. So thorough characterize and understand the quality of the batches used in your exhibit batches, stability, and also BE studies and everything. Quickly identify the root cause for inconsistent DPI performance and optimize product design at early phase of the development to minimize the risk. Also establish control strategy based on the evidence, scientific evidence gathered from thorough characterization of the stability batches and BE batches. At the end, I would like to acknowledge my office and my division, Division 5, my division director, Yi Ting, Deval Gaglani, my supervisor, and my colleagues in Unit 2. Also, I would like to acknowledge Office of Pharmaceutical Quality Research 2 in St. Louis, Jason, Rogers, Chining, and Nicholas. Office of Process and Manufacturing Assessment, Charmesta, Chining, Chu, and Vicky, and also OGD, Office of Research and Standard, Division of Therapeutic Performance 1, past and present members of nasal and inhalation product team. Thank you so much, and I'm looking forward for all questions. And I'm welcoming my other colleagues, my other speakers, and the panelists. Thank you.